Hello, Bio30, and welcome to the very first video on mutations. This is video 1 of 2. We're going to start off our time in point mutations. Today, your job is you are learning how to evaluate and demonstrate the consequences of mutations that happen within the body. We're going to talk about how we understand this through a couple different things. You need to be able to tell me what is a gene, or we also refer to them as point mutations. What are they compared to a chromosomal mutation? That's video 2. I want you to be able to tell me the difference between a frameshift and a non-frameshift mutation. And within that, there's 4 different types of mutations: additions, deletions, substitutions, and inversions. You should be able not only to compare the 4, but also demonstrate them through a different variety of activities, which we will do later. You should also be able to tell me why additions and deletions are more dangerous than, let's say, a substitution and inversion. The last 3 are going to be found in a separate video. So to start our time off, we're going to fill in the notes on page 8 of our note package. And essentially, it's just talking about where do the mistakes happen. So mistakes start off during DNA replication. So helicase unwinds, DNA polymerase comes in and is supposed to start putting nitrogenous bases, ACs, Ts, and Gs, onto complementary DNA strands. Now, a gene mutation, or from point mutation is what I'm going to call it from now on, essentially is when that doesn't happen. The DNA polymerase misses something, puts the wrong one in, or when it's fixing mistakes, it misses one. And what it does is it conserves that bad gene and that bad DNA, and it passes it on, and this error gets passed on and passed on and passed on. How does this even occur? This occurs through something called mutagens, things like X-rays or being exposed to UV rays or radioactive material. There's lots of reasons why, and sometimes they just randomly happen. So a quick aside, just to kind of help us round out this idea, we have not learned about transcription and translation yet. That'll be in next week's videos. However, what you do need to know is I'm going to use some terminology during this video, and I want to explain it quickly. We know DNA is made of nitrogen bases, ACs, Ts, and Gs. But what you may not know is we put them together in what we call triplets or codons. What that essentially means is that we take 3 letters and put them together, like ATT or GCG or ACT. And that is called a codon or a triplet, triplet, right? 3. Now, what is important about that is those triplets end up being what is read to make proteins in your body in the simplest terms possible. We will learn the actual process in transcription and translation. Now, during replication, this process of making these codons or triplets gets disrupted, and there's an issue with it that causes a point mutation. Okay, so one of those nitrogenous bases to accidentally get disrupted, and that is how we end up with a point mutation. So we're going to start into this chart here with 2 different types of mutations, frameshift and non-frameshift. What you have to understand is that a frameshift mutation will have an effect on all the DNA that comes after that mutation. So that little codon, that 3 letters, ATT, anything after that in a DNA sequence will also have an effect. In a non-frameshift mutation, only the 3 letters are affected. So ATT is the only one affected, nothing else. Within these 2 categories, you have additions and deletions on the frameshift side, substitutions and inversions on the non-frameshift side. I'm going to walk you through all 3 using an example. Please, you can write the example down on the side if you want to help you remember, or you can just write what's in the actual notes. So we're going to start in the frameshift side. And we're going to start with this character right here, the Beast. He's one of my favorite Disney characters. Belle is actually my favorite princess, so I just decided to use him. He's a great way to start this off. So we're going to refer to our original DNA sequence as the Beast, or just Beast, B-E-A-S-T. And what we're going to start with is talking about an addition mutation. So an addition mutation is when you add a letter into the sequence. So this changes what your original sequence was into something completely different. So if I'm taking Beast as an example, if I had the word Beast and I inserted an R, so we added the letter R, all of a sudden it becomes breast. This is not the same thing as before. I hope you recognize that. I added something in and I created an entirely different object. This example of the beast is one you can add to the side if you want, or you can leave it out completely. What I want you to add into your notes is this. So additions are extra bases added. For example, if the word beast we translated into DNA, it might be translated as AAC, TCT, AGG. Those little 3 sections, AAC, that's a codon, that's a triplet. We're going to pretend that this beautiful set of 3 codons codes for the protein for insulin. All of a sudden, we have an addition added into our DNA sequence. So rather than AAC, we now have AAA. We added an A into the second, inserted it into the second codon, the second base. So we added an A right into here. What that does is it shifts, or it causes the frames, these 3 letters here, to shift over one. So AAA pushes this C into the next frame, which then pushes this T into the next frame. And this G now is all by itself. It's shifted, hence why we call it a frame shift. Now the problem is once we did that, these lovely letters no longer code for the same things that this did. And we're going to learn how they code for proteins, but essentially just know they're not reading the same. So because they don't read the same anymore, now we don't code for insulin. And your body now can no longer produce insulin because it doesn't know how to, because the protein that normally did it is gone. So that's an addition. We're going to do this again. We're going to go back to our frame shift. This time, using the beast again. He's our original DNA sequence, but this time we're going to go through a deletion. So again, this is one of our nitrogenous bases, A's, T's, C's, or G's. Now instead of getting something added in, we're going to lose something. We're deleting it. So beast loses an A and now becomes best. Best of what? The best of something, but he's no longer a beast. So this is a problem because, again, it completely changes the overall structure of everything. A deletion is when you end up losing a base. A base is an A, C, T, or G. So again, there's my original sequence that codes for insulin. This time you can see that I lost a letter. I lost this T right here. This T disappeared. What it did is it shifted or frame shifted everything backwards. So now I have the original protein codon here, but this has changed and this has changed. With a deletion, more or less, it stops coding for any protein. But you can't code for anything if you only have 2 here. This isn't the same as anything. So again, this affects the overall DNA because it stops coding for the protein it was supposed to. So now you can kind of see why frame shifts can be very dangerous. These mutations cause massive changes to your DNA, so they stop functioning, they stop coding for a protein that makes up you. If you stop coding for insulin, we know what happens. That can result in diabetes. That means you'll be on insulin for the rest of your life, synthetic insulin, giving yourself shots. This is key to understand that a protein that makes up you, if we change it, can have massive effects. We're going to go back to this chart, except to the other side this time, and we're going to talk about non-frame shifts. This is when just the one of the 3 letters is affected. So we're going to go back to our example of the beast. He's our original DNA sequence still, and we're going to substitute this time. We're going to add a letter where there already is a letter. We're not going to add an extra letter. We're just changing out one letter. So beast substitutes in an F, and we get feast. This is not the same thing. Obviously, a beast and a feast are different. But the cool thing about our DNA is there's a lot of redundancy, a lot of repetition. So even though the B became an F, if we're lucky, that word can actually still function the same, depending on how the change worked. So here's an example of the substitution. I'm going back to my original codes for insulin. Now below AGG, you'll see I wrote the word serine. Serine is a type of amino acid that'll eventually make up a protein. We know amino acids are the building blocks of protein. We learned that in Bio 20 in the macromolecules section. So amino acids are the smallest portion of a protein. And this one's name is serine. AGG gives me serine. If I substitute a T for this final G, so right here, here's my G, I substitute it in a T, I still get serine. I still read AGT still makes me serine. Now this doesn't make sense yet because we haven't accomplished transcription and translation. It will. What I want you to understand from this is substitutions is taking one letter and changing it for another. So the unique thing about amino acid triplets or codons is that there's so much redundancy, guys. I've already said that. So a substitution doesn't always mean an issue. That's not always the case though, and I'll talk about when it's actually dangerous. But usually, okay, a substitution doesn't necessarily mean that there is a problem. Our very last one we're going to talk about, going back to my beast example, is an inversion. This is when the triplet switches itself up. So it stays a triplet, but the letters just jumble up a little bit. So for example, if I had beast, the T and the S switched place by accident, and now we have beets. Again, beets and beast are not the same thing, but all the letters are still there. So it could code for the same thing. An inversion is a single DNA triplet that inverts itself. So I've already given you this in your notes and you can already see it. Hopefully you can see that it's the middle section of codons that are changed. So what does this mean? Non-frame shifts. Most of the time they occur in what we call non-coding DNA. You don't need to know this at the Bio 30 level, but essentially there's a lot of DNA that we have that codes for absolutely nothing. And so if you have an issue in that area, you're not going to have any problems whatsoever. However, sometimes substitutions and inversions cause a change up, but the amino acid doesn't change. If the amino acid doesn't change, then that means it codes for the same protein. Now, there's always an exception to this rule. Sometimes when you get an inversion or a substitution, something does happen. A lot of diseases that we know, such as sickle cell anemia or color blindness, maybe you've heard of Tay-Sachs, are actually point mutations that have occurred. Essentially, a substitution has happened that causes this disease to arise. So there is sometimes a danger to a non-frame shift mutation. So I hope you have these all in your notes and you're kind of understanding. Please do the example underneath the summary points. Try it out. I will post an answer key to the example later on.